Thank you for that introduction, Leon. Good afternoon, everyone. My name is Sachin, and I'm excited to present our Deshpande project titled Bottlebrush Prodrugs, a platform for controlled modulation of the immune system. Immunotherapies have shown promising results in a subset of patients in the context of long-term progression-free survival, with 50% of all cancer patients in the US today being treated with currently available immunotherapies. Thus, there is a huge push towards developing novel approaches rooted in immunotherapy, with over 1,500 clinical trials, either as a monotherapy or in combination with standard treatments such as chemotherapy and radiation therapy. One such approach is the use of small molecule immunomodulators, such as resiquimod, which have shown incredibly promising anti-tumor effects. Now, the catch with these molecules is that even though they are incredibly potent and activate the immune system against cancer, they're extremely toxic, especially if you give it systemically in the blood, since it results in a cytokine storm and patients have reported flu-like symptoms. So currently, all small molecule immunotherapies are administered either topically or intratumorally in tumors that are accessible. And while this is an incredibly promising approach, a potential shortcoming with this is that you're relying on immune cells, such as T cells, to go to unmodulated tumors which might not be as responsive. And 90% of cancer patients die not because of the primary tumor, but because it has metastasized throughout the body. And so systemic immunotherapy could be an incredibly promising approach to change the microenvironment in all tumors simultaneously. So that's the essence of this project, is can we develop an approach that provides sufficient release of these immunotherapies in tumors to demonstrate efficacy, while slow enough release in the blood and other organs to lower systemic toxicity. Metaphorically speaking, think of it as a delivery vehicle that preferentially releases its cargo once it reaches the tumor with minimal release in the blood. In order to achieve this, we have developed a brush platform where we synthesize drug-loaded individual units and then using a technique called ring opening metathesis polymerization, we combine them into bottlebrush structures. This strategy allows us to tune both the size as well as the release rates of the immunotherapy to find a balance between maximizing the performance and minimizing the toxicity. So we started off with one particular combination with size 10 and the slowest releasing linker. And we did an MTD study, a maximal tolerable dose study, to understand with the free drug, how much can we load in mice without seeing signs of toxicity. And we saw that we could load about 7.5 mg per kg. Whereas with our BPD platform, we could go 10 times higher without any signs of toxicity. And we also saw that when we injected it in tumor-bearing mice, the BPD containing the drug resiquimod R848 showed a much stronger anti-tumor effect as opposed to the free drug. And we also saw, finally, that it is effective as a combination therapy with an FDA-approved antibody referred to as anti-PD-1, where again, when the anti-PD-1 was delivered with our R848 BPD, we saw a strong anti-tumor effect as compared to the antibody alone or the free drug and the antibody. So the next steps include trying multiple different sizes to further improve the efficacy while lowering the toxicity, delve deeper into organ and cellular immune interactions of the BPDs, and finally test it with multiple immunotherapies to develop this patented platform technology with the goal of licensing it to pharmaceutical and biotech companies facing toxicity issues with their own treatment approaches. Finally, I'd like to acknowledge my mentors and the team that helped with this project, as well as the Deshpande Center for their support. Thank you so much for listening.